Welcome to Biologics. In this tutorial we are going to learn about biological classification. This tutorial is helpful for students of Class 11, the Two-Kingdom classification. The Greek philosopher Aristotle is also known as the father of biology. He developed the first widely accepted system of biological classification. He classified the living world into two kingdoms, Kingdom Animalia and Kingdom Plantae. Animals were further grouped based on absence or presence of red blood, into bloodless and red-blooded. This distinction of animals nearly matches the modern distinction of invertebrates and vertebrate. However, this system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, or between photosynthetic and non-photosynthetic organisms. Consequently, a large number of organisms did not fall into either category. Hence the two-kingdom classification used for a long time was found inadequate. Five-kingdom classification. Due to the shortcomings of the two-kingdom concept, a need was felt for including features besides gross morphology, for example, characteristics like cell structure, nature of cell wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. R. H. Whitaker in 1969 proposed a five-kingdom classification. The kingdoms defined by him were named Monera, Protista, fungi, Plantae, and Animalia. The main criteria for classification used by him includes cell structure, thallus organization, mode of nutrition, reproduction, and phylogenetic relationships. The earlier systems of classification included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and the angiosperms under plants based upon the presence of cell wall. Consequently, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group, fungi, and the autotrophic green plant. Now let us compare a few features exhibited by members of each kingdom. Cell type. Members of Kingdom Monera have prokaryotic cell types while the other kingdom has eukaryotic cell types. Cell wall. Monerans have a non-cellulosic cell wall composed of polysaccharides and amino acids. In some protists cell wall is present, while in fungi the cell wall is without cellulose. In Plantae, the cell wall is present and made up of cellulose, while cells of the members of Kingdom Animalia do not possess cell walls. Nuclear membrane. Except the members of Kingdom Monera, cells of the members of all other kingdom have nuclear membranes. Body organization. Members of the Kingdom Monera and Protista show organization at a cellular level, while the members of fungi show organization at the multicellular and loose tissue level. Members of the Kingdom Plantae and Animalia show tissue and organ level of organization. Mode of nutrition. Monerans exhibit a diverse range of nutrition ranging from chemosynthetic autotrophs to heterotrophic nutrition including saprophytic and parasitic mode of nutrition. Members of the Kingdom Protista are either photosynthetic autotrophs or heterotrophs. Members of Kingdom Fungi are either heterotrophic saprophytes or parasites. Members of Kingdom Plantae are photosynthetic autotrophs, while the members of Kingdom Animalia are obligate heterotrophs exhibiting holozoic or saprophytic mode of nutrition. Kingdom Monera. Bacteria are the sole members of the Kingdom Monera. They are the most abundant group of microorganisms. Bacteria occur almost everywhere including extreme habitats such as hot springs, deserts, snow, and deep oceans where very few other life forms can survive. Bacteria show extensive metabolic diversity and many of them live within, and, on other organisms as parasites, symbionts, and commensals. Bacteria are grouped under four categories based on their shape: the spherical coccus, plural, cocci, the rod-shaped bacillus, plural, bacilli, the comma-shaped vibrium, plural, vibrio, the spiral spirillum, plural, spirilla. Kingdom Monera is further divided into two large groups. Archaeobacteria. These bacteria are special since they live in some of the harshest habitats such as extreme salty areas, for example, halophiles, hot springs, for example, thermoacidophiles, and in marshy areas, for example, methanogens. Archaeobacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Eubacteria or true bacteria are characterized by the presence of a rigid cell wall, and if motile, a flagellum. Eubacteria includes cyanobacteria, also referred to as blue-green algae. Cyanobacteria are photosynthetic autotrophs and have chlorophyll A. Chlorophyll A of cyanobacteria is similar to chlorophyll of green plants. Chemosynthetic autotrophic bacteria. These bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia and play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria. These bacteria are the most abundant in nature, and the majority of them are important decomposers. Many of them have a significant impact on human affairs such as curd making, cheese making, etc. Mycoplasma. Mycoplasmas are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasmas are pathogenic to animals and plants. Kingdom Protista. All single-celled eukaryotes are placed under Kingdom Protista. The members of Protista are primarily aquatic. This kingdom forms a link with the others dealing with plants, animals, and fungi. Protists being eukaryotes, the protist cell body contains a well-defined nucleus and other membrane-bound organelles. Some protists have flagella or cilia for locomotion. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. Protists are further divided into chrysophytes, dinoflagellates, euglenoids, slime molds and protozoans. Chrysophytes. This group includes diatoms and golden algae, for example desmids. They are found in fresh water as well as in marine environments. They are microscopic and float passively in water currents, for example plankton. Most of them are photosynthetic. In diatoms the cell walls form two thin overlapping shells, which fit together as in a soap box. The cell walls are embedded with silica and are indestructible. Thus, diatoms have left behind a large amount of cell wall deposits called diatomaceous earth used for polishing, filtration of oils and syrups. Diatoms are the chief producers in the oceans. Euglenoids. These are freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicle which makes their body flexible. They have two flagella, a short and a long one. They are photosynthetic in the presence of sunlight. When deprived of sunlight they behave like heterotrophs by predating on other smaller organisms. Slime molds. Members of this group are saprophytic protists. Their body moves along with decaying twigs and leaves engulfing organic material. Under suitable conditions, they form an aggregation called plasmodium, which may grow and spread over several feet. Under unfavorable conditions, the plasmodium differentiates and forms fruiting bodies bearing spores at their tips. The spores possess true walls and are extremely resistant and survive for many years, even under adverse conditions. The spores are then dispersed by air currents. Protozoans. All protozoans are heterotrophs and live as predators or parasites. They are further divided into four subtypes. Amoeboid protozoans. Amoeboid protozoans live in fresh water, seawater or moist soil. They move and capture prey by pseudopodia. Example, amoeba. Flagellated protozoans. The members of this group are either free-living or parasitic. They have flagella. The parasitic forms cause diseases such as sleeping sickness. Example, trypanosoma. Ciliated protozoans. These are aquatic, actively moving organisms because of the presence of thousands of cilia. Example, paramecium. Sporozoans. This group includes diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, the malarial parasite. Kingdom fungi. The fungi constitutes a unique kingdom of heterotrophic organisms showing a great diversity in morphology and habitat. Fungi are cosmopolitan, and occur in air, water, soil and on animals and plants, and they prefer to grow in warm and humid places. With the exception of yeasts, which is unicellular, fungi are filamentous. Their bodies consist of long, slender, thread-like structures called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes, filled with multinucleated cytoplasm. These are called cenocytic hyphae. Others have septae, or cross walls, in their hyphae. The cell walls of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophic, and absorb soluble organic matter from dead substrates, and hence are called saprophytes. Some of them depend on living plants and animals and are called parasites. Some can also live as symbionts, in association with algae as lichens and with roots of higher plants as mycorrhiza. Reproduction in fungi can take place by vegetative means, such as fragmentation, fission and budding. Asexual reproduction occurs by spores called conidia or sporangiospores or zoospores, and sexual reproduction occurs by oospores, ascospores and basidiospores, produced in fruiting bodies. The sexual cycle of fungi involves the following three steps. 1. Fusion of protoplasms between two motile or non-motile gametes. This process is called plasmogamy. 2. Fusion of two nuclei, known as karyogamy. 3. Meiosis in zygote resulting in haploid spores. Depending on the morphology of the mycelium, mode of spore formation and fruiting bodies form the basis for the division of the kingdom into various classes. Phycomycetes. Members of Phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places or as obligate parasites on plants. Their mycelium is aseptate and cenocytic. Asexual reproduction takes place by motile, zoospores or by non-motile planospores, endogenously produced in sporangium. Zygospore is formed by fusion of two gametes. These gametes are similar in morphology, isogamous or dissimilar anisogamous or oogamous. Example, mucor. Ascomycetes. Members belonging to ascomycetes are commonly known as sac fungi. The ascomycetes are mostly multicellular, for example, penicillium, or rarely unicellular, for example, yeast, Saccharomyces. They are saprophytic, decomposers, parasitic or coprofilous, growing on dung. Their mycelium is branched and septate. The asexual spores are conidia produced exogenously on the special mycelium called conidiophores. Conidia on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac-like asc, singular, ascus. These asc are arranged in different types of fruiting bodies called ascocarps. Example, Aspergillus, Basidiomycetes. Members belonging to Basidiomycetes are commonly known as mushrooms, bracket fungi or puffballs. They grow in soil, on logs and tree stumps and in living plant bodies as parasites, for example, rusts and smuts. Their mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotypes. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis take place in the basidium, producing 4 basidiospores. The basidiospores are exogenously produced on the basidium, plural, basidia, arranged in fruiting bodies called basidiocarps. For example, agaricus, Ustalago, and Puccinia. Deuteromycetes. Members belonging to Deuteromycetes are commonly known as imperfect fungi because only the asexual or vegetative phases of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they rightly belonged to and were correctly identified and moved out of Deuteromycetes. The Deuteromycetes reproduce only by asexual spores known as conidia. Their mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria, Colototricum, and Trichoderma. Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll-containing organisms commonly called plants. A few members are partially heterotrophic, such as the insectivorous plants or parasites. For example, bladderwort and Venus flytrap are examples of insectivorous plants, and Cuscuta is a parasite. The plant cells have a eukaryotic structure with prominent chloroplasts and cell wall, mainly made of cellulose. Kingdom Plantae includes algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Life cycle of plants has two distinct phases, the diploid sporophytic, and the haploid gametophytic, that alternate with each other. This phenomenon is called alternation of generation. Kingdom Animalia. This kingdom is characterized by heterotrophic, eukaryotic, multicellular organisms, and their cells lack cell walls. Members of Kingdom Animalia are directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserves as glycogen or fat. Their mode of nutrition is holozoic, by ingestion of food. Animals follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanisms. Most of them are capable of locomotion. Sexual reproduction occurs by the copulation of male and female, followed by embryological development. Viruses, viroids, prions and lichens. Viruses did not find a place in classification since they are not truly living, if we understand living as those organisms that have a cell structure. The viruses are acellular organisms that are characterized by having an inert, crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. The name virus, meaning venom, or poisonous fluid, was given by Pasteur. D.J. Ivanowski in 1892, M.W. Bajer-Inuk in 1898, demonstrated that the extract of the infected plants of tobacco could cause infection in healthy plants, and called the fluid as contagium vivum fluidum, infectious living fluid. W. M. Stanley in 1935, showed that viruses could be crystallized, and crystals consist largely of proteins forming a protein coat called capsid. Viruses are obligate parasites, but they are inert outside their specific host cell. Viruses also contain genetic material, that could be either RNA or DNA. No virus contains both RNA and DNA. Viroids. In 1971, Theo Diener discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein coat that is found in viruses, hence the name viroid. Prions. Certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded proteins. The agent acted similar in size to viruses, and was called prions. The most notable diseases caused by prions are bovine spongiform encephalopathy, BSE, commonly called mad cow disease in cattle. Lichens. Lichens are symbiotic associations, and are mutually useful associations, between algae and fungi. The algal component is known as phycobiont and fungal component as mycobiont, which is autotrophic and heterotrophic, respectively. Algae prepare food for fungi, and fungi provide shelter and absorbs mineral, nutrients, and water for its partner. Lichens are very good pollution indicators, as they do not grow in polluted areas. Thank you for listening. For more educational content on various subjects of biology, genetics, immunology, molecular biology, do visit Biologics, and don't forget to like and share our videos. See you next time.